Never complain about the things your parents could not give you. It was probably all they were having and they were just able to give it to you. So the bacterial genetics continuing. Number two is transduction. It is the gene transfer from a donor to the recipient by abnormal bacteriophage. Bacteriophage is a virus that infects bacteria. Phage coat protects the DNA so resistant to the environment. Nucleases cannot be destroyed by these enzymes. Infectious agents that replicate as obligate intracellular parasite in bacteria. Significance is it is common in gram-positive lysogenic phage conversion, e.g., Corynebacterium diphtheriae toxin, when it is being transferred through the bacteriophage, so these become infective in nature and causing the problem. Its extracellular phage particles are metabolically inert. They are different types of the phage, polyhedral, filamentous, and complex in shape. Not all phages can do transduction. Few are capable of doing that. In bacteriophage, either DNA or the RNA can serve as genetic material. Phage structure, if you just see it, it is the structure of the T4 bacteriophage, that is the head, that is the tail, contractile sheath, tail fibers, and the base plate by which it do attach and transfer by the sheath contraction the genetic material which is here in the head to the bacteria on which it have got the attachment by the tail fibers and the base plate. So the different components of the phage are head of the capsule, tail, and the adsorption or just attachment to the bacteria on which it is going to transfer the genetic material via that adsorption, that is by tail fiber. Receptor is LPS for T4, irreversible attachment, and base plate by which it get attached. Here one can see the phage composition. It is having the nucleic acid, protein, protection, infection by these proteins, sheath contraction and transfer the genetic material, nucleic acid injection, and the DNA uptake by the bacteria. Here one can see the bacteriophage and that is infecting the bacteria, and one can see the bacteriophage is transferring its genetic material into the bacterium, and then the bacterium is going to produce the more bacteriophage which are capable of just infecting the other E. coli bacteria, and then these are all being infected by these bacteriophage, and ultimately that is going to infect the new bacteria. Now the types of the transduction generalized here, any bacterial gene or the donor can be transferred. Specialized, as the name is, only certain specific genes of the donor can be transferred. Abortive transduction, the transient expression of the donor genes without formation of the recombinant progeny, and the complete transduction, production of the stable recombinants. So, generalized transduction, it is the infection of the donor. Potentially, any donor gene can be transferred. Phage replication and degradation of the host DNA, assembly of the phage particles, and infection of the recipient. Here one can see the infection of the donor, and then this chromosome is going to be disintegrated, and ultimately this is being occupied and taken up by the phage. These phages are now replicating inside the host cell, and as you can see, these are just releasing the host cell by the lysogenic or by the other process in which the host cell is destroyed, and the released these just bacteriophage are capable of infecting the new bacterium. As you can see, the first one was just being attacked, and ultimately so many bacteriophages have been produced by this process of transduction, generalized transduction, and capable of transfer. Now, the pore are so big bellies that the rich are so poor, big bellies that the pore just comes under the belly, and the rich man is not able to see where the pore is just lying. The other process is the conjugation. Here one can see the gene or the DNA is transferred from a donor to a recipient by the direct physical contact, that is called bacterial sex. Here is formation of the cytoplasmic connection between the donor and the recipient bacteria, and this is called mating types in the bacteria or the bacterial sex. Donor has F factor or the fertility factor and F sex pilus, and that is in bacteria. There are two mating types, a donor male and the recipient female, and here direction of the transfer of the genetic material is only one way, that is from the donor towards the recipient, not from the recipient to the donor. Here one can see the mating or the bacterial sex, the chromosomal DNA. As you can see, the plasmid through the pilus, that is the connection between the two bacteria, and that is being transferred. The genetic material, the plasmid, is being transferred to the other bacteria. Here again, one can see the actual sex pilus, which is transmitting and transferring the genetic material to these cells. And the conjugation, that is having a donor, a bacterium said donor, as has an extra piece of the DNA called F factor or fertility factor or sex factor, which that bacteria can donate, so that is called donor. Now, a simpler piece of the DNA that can replicate itself in the cell, that is called replicon. There are usually plasmids which are taking part in the conjugation, and the F factor has genes on it that are transferred to a recipient through a sex pilus. F factor has ability to produce a sex pilus, F pilus, on the surface of the bacterium, through which it can transfer the genetic material to that. The pilus is important in the conjugation process. Here one can see the bacterium which is having the F factor, that is just going to transfer to the other which is not having the F factor. So this F factor positive can be transferred to the F factor negative through this F pilus, and then these are having the same, and now they are again capable of transferring the genetic material from one bacteria to another, because now this has got the F factor, it has become the F positive, it was already F positive. So these are the donor cells, and the recipient cells become the donor again. Here one can see the chromosomal DNA and the F plasmid. The donor is having S plasmid, and through that pilus it is going to transfer it to the recipient, and here one can see the sex pilus is combining with this recipient, and the DNA polymerase enzyme, rexosome, and other genes can be transferred to this chromosome, and when it has transferred this has become a donor. That was the old donor which has transferred this. Now the new donor that has got the plasmid, F plasmid, now it is capable of just transmitting that to the other. So recipient is that the bacteria which act as recipient is without F factor. Strains of the E. coli with an extra chromosomal F plasmids are called F positive. It function as donors, whereas strains that lack F plasmids are F negative. It behave as recipient. Significance in gram-negative bacteria, antibiotic resistance is causing the rapid spread. In gram-positive bacteria, production of the adhesive material biofilm by the donor cell is also transferred through the conjugation to the other cell. Here one can see bacteria do attach and they are going to have the maturation, and after maturation they are going to have the attachment and to make the biofilm again, the new one there. So the summary of all this is that transformation, the bacteria take the naked DNA pieces in the environment and they are just being taken by the competent bacteria. Here the phage is going to transfer the genetic material to the one bacterium and that multiply the bacteria phage multiplied in that bacterial cell, and when it disintegrates, this is going to infect the other bacterium. And here in conjugation, one bacteria through the pilus and can transfer the genetic material from one bacteria to another. So that is a kind of bacterial sex or the conjugation. So that was the summary. Here one can see again these are the wild or the naked type of the genetic material or the chromosome lying in the environment, and that is being taken up by this bacteria as part of that naked DNA, and that is going to be merged with this chromosome. Here that plasmids are being transferred from this, that is the piece of the genetic material or the plasmid as a whole being transferred to this bacterium, and that is just going to merge with chromosome, that is the sex pilus, or that is the conjugation process. That is transformation, the naked or genetic material taken up by the bacteria, and here there is transduction. The bacteriophage is going to inject the genetic material to the bacterium, and that genetic material injected by this bacteriophage can be taken up by the plasmid, or it can merge with the chromosome to make these bacterium more competent and with the resistance genes that is being transferred from this plasmid, from this bacteriophage, or from these wild naked pieces of DNA. So that is the end of the lecture, and I hope and request you are going to subscribe this. If you like, so you can share it with others, and just if any question or the query, never hesitate to ask me, and I will be happy to answer. And thank you very much for watching.